I have the great pleasure of introducing our speaker, who's also a good friend of mine, Mary Cain, from Kansas State. She did her PhD work at the University of Vermont before going on to do a postdoc at the University of Kentucky with Mike Bardo. So that's sort of where the 1 degree of separation between our training occurred. She has now been on faculty since that postdoc at Kansas State University, and she's got a very accomplished record of publications. She's a project leader on a P20. One of my favorite grant mechanisms is the R15. I love those grants. She's been continuously funded on R15 from 2013 to 2021, where it continues on differential rearing and glutamate and addiction. So you can just keep going with that topic for like, it's so broad. It's an awesome title. It's a great title. Yeah. She is a fellow of the American Psychological Association for 2 divisions, Division 6, which is Behavioral Neuroscience and Comparative Psychology, as well as Division 28, which is Psychopharmacology, and I should know, I was former president of it. But you were president recently of Division 6, 2018. What was the last, I think that was your post. We had to still be on the executive committee. She's on the editorial board for Behavioral Pharmacology, which is one of our favorite journals to go to. And she's also does extensive reviewing for NIH. So if you please join me in welcoming Mary.
Thank you, Rick, for a very nice introduction. And thank you for coming out on this Thursday afternoon. As Rick mentioned, I'm down at Kansas State University, so I'm about a little over 2 hours south. It's an easy drive. For those of you that have not been to Manhattan, I think Kansas State, we're well known for our athletics. We used to have this football coach, Bill Snyder, you may have heard of him if you're into athletics, Willie the Wildcat. We have a new coach, Chris Kleinman. People care in Manhattan. You may not care as much here. Anyway.
We recognize the theme.
Okay, yeah. So there's a lot happening, bowl selection, everything. But we actually, we do have academics at K-State also, which we're very proud of. We have a beautiful native limestone campus. This is our historic library. It's awesome. And I'm in psychological sciences. We're right across from this beautiful building. Unfortunately, we were built during the energy crisis, and Rick has seen our building. Yeah. The good news is we have no windows, so we can't see how beautiful campus is.
No distractions.
Yeah. And we're all set for the zombie apocalypse because there are no windows. Yeah. And I'm in the behavioral neuroscience area, and we have a rat lab, so we always joke too, we have food. We have rat food. We have all the food for the rats and water and stuff. So we're set for a couple of months with our Purina rat chow if the zombie apocalypse happens. So anyway, no, we have I'm in the psychological sciences. We have a graduate program, and within that, I'm in the behavioral neuroscience division. So no zombie apocalypse. All right. And I work with a great group of undergraduate and graduate students who I definitely need to acknowledge. We're having a mock Thanksgiving dinner. We actually got this idea from Rick when he came to visit. My students love that idea. So Rick, do you still do that?
Practice Thanksgiving?
Yeah, it does a practice Thanksgiving. Students can practice their dishes. So we kind of took your tradition and spun it a little bit, and we do this every Thanksgiving. So there we are, having our mock Thanksgiving. And I work with several great people, including a former faculty member of my department who's now emeritus, and he's loving it, Stephen Kiefer, faculty member over in chemistry, Stefan Bosman, trying to figure out where my mouse is. Anyway, there it is. All right, and then a former grad student of mine, Eric Garcia, who's now a postdoc at UT Galveston. Current grad student who I'll talk more about a little bit, TJ Wukich, and then Ted and Troy. We'll talk about them in a little bit, and a bunch of wonderful undergraduates as well. And then I'm funded by NIDA, and as Rick mentioned, P20, that's our co-brace center, and our co-brace center is called CNAP. So that's where I'm fortunate enough to have my funding come from. And as mentioned by Rick, and title of my talk, I'll be talking about the effects of environmental enrichment. So I'm really interested in how does the early rearing environment alter behavior and this term incentive motivation in adulthood. And incentive motivation is put simply, the liking and wanting of reinforcers. So how much do you like and want the reinforcer? And I'm looking in adulthood. But first I'll talk to you about the environmental enrichment. I use a well-established model of differential rearing that was developed back in the 1960s. This is a picture I borrowed from the New York Times. This is Rosenwig, Diamond, and Bennett in 1964, looking in at their enriched environment, and they developed the environmental enrichment model, and then others, including Greenow, who you may have heard of, have done extensive work in this area, demonstrating that novel objects, handling, contact with a cohort during early childhood, adolescence has profound effects on the brain and behavior in adulthood. And I would argue this early work, it had a large impact on learning and memory, but it also had a large impact on our policy. It's, I think, some of the foundation of things like Head Start, knowing that we need to enrich that early environment, that it impacts adulthood. And they did some really great work. So my model is based on their model and also based on my mentors like Mike Bardo at the University of Kentucky. But a lot has changed in the animal care and youth world since 1964. So while I still use that model, we've updated it for current IACOC approval. So this is what my enriched condition looks like today. Okay, so plastic objects, for example. Okay, so we have novel objects that get changed daily. The animals are raised in large groups, anywhere from 8 to 12 per cage, and they get handled daily by us. They arrive in our lab at 21 days of age. That's right after they wean. It's early childhood. And they remain in their conditions until postnatal day 51, which is meaningless. But for a rat, that's late adolescence, early adulthood. So right at the end of adolescence is when their rearing period ends. And then we start all training and testing. We have done some studies where we switch their environments, but everything I'll be talking about today, they stay in their environment all the way through. So there's no switching. We start testing late adolescence, early adulthood, but they stay in their environment all the way through the experiment. This is in stark contrast then to the isolated environment. We use the, this actually used to be the standard of care for rodent cages. These are old cages that were in our facility. We do not handle the rats for the 30-day rearing period. They have no contact with their cohort and they have no novel objects. But really important to note, we do monitor their food and water intake daily, and if we see any changes in food and water intake, we do handle them, we do health checks, and proceed accordingly. But healthy animal does not get handled for the 30 days. So there are numerous differences between these two environments, and those of you with a psychology background, if you start thinking about control groups, you might actually start to get a headache because you have differences in cage size, bedding, novel objects, handling, cohorts. I think there is a total of 9 different elements that differ between the two environments. And running an experiment with 9 different control groups just was not productive. One cannot make any forward progress in one's research program. So what I do and what other researchers do is we often include a standard condition. And this is just 2 rats per cage that are handled once a week during bedding changes, and this adheres to the National Institute of Health guidelines. It is by no means a control group for all the differences. It's just an average rat. So we have an average rat. What does the average ordinary rat do in our lab? And then we have the 2 contrasting environments, but it by no means controls for all the differences between the 2 environments. We don't try to do that. It would stop us in our tracks, having all the different control groups. So this is how we manipulate the early rearing environment. And as I mentioned, that early rearing environment has profound changes on synaptic plasticity, on the brain, and on behavior. And the behavior I'm interested in, again, is incentive motivation, liking and wanting, and specifically of drugs. So to ask that question, big change, forgot this was my next slide, but to ask this question, we do give our animals drugs. We use intravenous self-administration, really similar to what Rick is doing. We implant an indwelling jugular catheter in our rats using anesthesia and surgery. And then once they recover from surgery, we place them in the operant chambers and they learn to lever press. Okay, so they learn to press a lever and they get an infusion IV of whatever drug we're studying. And then we always have another lever in the chamber that they can press all they want, but nothing happens. And we always have that in there to be sure they're really pressing for the drug on the active lever. So we always have a control lever in there as well. So just to give you orientation, okay, and we have cue lights, make that really obvious. Just to give you some orientation, we have a pump up here with the drug, and then they're trained to lever press on the schedule, and when they lever press, they get an infusion of the drug, and the tubing is attached to them where the drug comes in, and then those drug infusions, we can pair them with cue lights, we can pair them with tones. So not only can we look at drug effects, but we can also look at Pavlovian conditioning effects as well of the drugs and of the chamber associated with the drug. And this is considered in preclinical models to be the gold standard, and that the rat gets to choose to take the drug using this model. The rat can choose to be in there and choose not to lever press, much as the human can choose not to lever press, or the rat can choose to take the drugs. And the majority of the drugs that we choose to use, they also choose to use. So it's a very good model of human drug-taking behavior. And it also is a great model using the animals here because it solves a lot of ethical issues. I was studying drug taking in late adolescence, early adulthood in drug naive animals. I cannot do that work in people. I cannot give drugs to people who've never had drugs in late adolescence, early adulthood and see what happens. So the animal model offers us a lot of opportunities that we can't easily do in humans. So it's a very good animal model of human drug taking. Good so far? All right.
Great.
So moving on then, mostly what I do, what my team does, we induce pharmacological or environmental manipulations. We then measure drug taking using that lever pressing as our dependent measure. And we also sometimes measure drug seeking, where we take the drug away and we see how much do they lever press just for the cues associated with the drug. And most of what I'll be talking about today is using this simple operant model for the first part of the talk. Okay. So when compared to isolated conditioned rats, enriched conditioned rats self-administer less psychostimulants. And psychostimulants include cocaine, amphetamine, methamphetamine. They will, in that chamber, lever press. Some chambers also have a nose poke. They'll nose poke, lever press, nose poke less than isolated rats. And it's a consistent finding across many different labs. And in terms of thinking about bigger picture, in terms of motivation, we consider this the wanted component of motivation. They self-administer less, and we think, therefore, they want the drug less. They're less willing to work for it. And in general, across labs, this effect is most robust with low unit doses of psychostimulants. So as we increase the dose, that protective effect enrichment becomes less robust. And I'll show you some data to explain this better. So this is a dose response curve for amphetamine. This is an increasing dose of amphetamine. This is a moderate dose, a lower dose, really low dose. And this is actually saline substitution. There's no drug, they're just getting saline. The isolated rats are in blue, the enriched conditioned rats are in red. And you can see at these lower doses, the isolated rats self-administer more than the enriched conditioned rats. But as we increase the dose, that protective effect of enrichment is no longer present. And to show you this, what happens at higher doses more clearly, this is repeated sessions at a high dose of amphetamine. 0.1 is a pretty nice high dose. Most animals will work for it. These are the number of times they press the active lever to earn the amphetamine. And now in this graph too, we have our average rats on there, the standard house rats. These are 10 one-hour sessions, and you'll see there's really no difference in lever press behavior. They press at a pretty steady rate. All three groups lever press relatively equally for the high dose of amphetamine. There's no protective effect of enrichment as we increase that dose. And this does beg the question, is enrichment protective? If it doesn't protect against these high doses, is this a good animal model? Should we be pursuing it as a protective model? I would argue it still is a good animal model because most individuals in late adolescence and early adulthood that start taking drugs don't start with these high doses. They start their recreational drug use with those low to moderate doses where we do see the protective effect. But this is concerning, obviously, as one gets into later stages of drug taking, it looks like enrichment may not be protective. So what a lot of groups are doing, these are 60-minute sessions, and that's good, but what a lot of groups are doing now is they're doing longer operant sessions to better model human behavior, because most drug takers for methamphetamine and meth, they don't just take their drugs for an hour and go home. They take their drugs in a binge pattern over several days for a long period of time, and then they take a break. So a lot of groups are doing what's called extended access, where they give the animals 6 hours a day, 8 hours a day. Some groups are even doing 22 hours a day, and to give them, to try to better model that binge with psychostimulant use. So we wanted to see if we did extended access. First, what would happen with amphetamine? Because normally, with extended access with cocaine, for example, you normally see it's really interesting. Here, they're really steady with a 1 hour session. With extended access, they actually increase over time, and they start to compulsively take the drugs. So we wanted to see with amphetamine, would compulsive drug taking develop, and would enrichment protect at a high dose with extended access? So we did 6 hour sessions with our high dose of amphetamine. We did 12 sessions. And these are the enriched, again, I tried to keep the color scheme, enriched are red, isolated, and standard house rats. And what we're seeing is enrichment is protecting against the escalation of drug taking across the repeated sessions. And then we're seeing relatively similar drug taking between the isolated and standard house rats. This study is ongoing. We actually just collected the last cohort 3 days ago, so I haven't run the stats, so you'll notice I'm saying it appears. But it looks like we have a protective effect of enrichment, and it looks like isolated and standard house rats are showing some escalation with amphetamine. So this is exciting. It does look like with extended access, enrichment may be protective even with the higher doses. And we're excited about this. And importantly here too, we had male and female rats. We saw no sex differences here with the repeated training with the extended access. Yeah.
So when they're not in session, are they removed from the?
Yeah, in our model, we remove them and put them in this study, they're put back in their respective environment. So the enriched rats are going back to their land of toys and novel objects and cohort and everything. So, and that's definitely, not to take your question to another place, but it's definitely something we would really like to do is what would happen if they went back to single housing for those 18 hours? Would we see that same protective effect now that we're seeing this? But we haven't gone there yet at all.
Could you say something about the saline? Why is there separation between EC and IC?
Yeah, that's a great question. How come the IC rats are lever pressing at a high rate for saline? In that study, the drug infusions are paired with a light, and IC rats are pressing for the light paired with the drug. They readily lever press for the Pavlovian conditioned drug cues. As Rick is smiling because he, yeah, there's a lot of work with the lights. Yeah.
Sensory stimulation on the isolated animals too.
Yeah, they will. Yeah. Yeah. Good question. All right. Good. Ooh, I went the wrong way. Okay. Another question we're really interested in is another phase of drug taking. So we're interested in the drug taking itself, but as many of you in this room know, many individuals who choose to seek help for their drug taking, they go to detox, go to rehab, whatever, and many people relapse when they return back to their home environment at alarmingly high rates. So there are many animal models out there for relapse, and we would really like to know, is enrichment protective against relapse? That would be wonderful if it was. So what we do for our relapse test is we give that extended access or 1 hour sessions or 6 hour sessions, prolonged exposure to amphetamine or drug of choice. They have a period where they have no exposure to the drug or the operant chamber. They remain in their respective home conditions for a period of time. Different labs do different things. You may do 2 weeks, you may do a month. We often joke in our lab, it's however long their insurance will cover, but it's more than that, but sorry. Anyway, we then put them back in the operant chamber on their test day, on their relapse test day. And importantly, when they go back in the same operant chamber now, after their break period, they have no drug available. So all the cues that were associated with the drug are present, but there's no longer drug available. So on the relapse test, again, we now have that famous saline is in the pump, no drug is present, the levers are present, and then those condition cues that were present with the drug, those are present, but the animal doesn't get any drug. And we just see how much do they lever press for the cues previously associated for the drug after the break period. And there are different ways to test this relapse or incubation, but this we were just looking at relapse. So we did 1-hour training sessions, and there was no difference in drug history during training. They got the same amount of amphetamine exposure during those 1-hour sessions. They then had a 10-day break period, or 10-day abstinence period, and then we looked at their relapse, and we saw that isolated rats showed the greatest amount of active lever pressing. Then our standard housed rats and our enriched rats did not show a significant increase, but it was close, in active lever pressing, suggesting that enrichment may protect or at least attenuate that relapse, which is exciting for us. So it not only protects against the drug taking phase, but it appears to also protect against relapse.
Is this in the 60-minute or 6-hour?
This is 60-minute, and actually later on I'll show you some 6-hour data. All right. So just to summarize, that was a lot. Enrichment is decreasing during those short 60-minute sessions at low unit doses predominantly. It's also decreasing, it looks like, with our newer data with extended access, those longer 6-hour sessions. It's also decreasing cue-induced relapse, and in general, again, coming back to that broader theme of incentive motivation, it's decreasing overall wanting for the drug at different phases of drug taking. So our group and many other groups have demonstrated similar results. And what we're really interested in, we love our behavioral data, but we really want to understand what about that early rearing environment, what's changing in the brain of these rats to produce this nice protective effect. Because if we can start to tease that apart, perhaps we can apply that to those who are vulnerable. So I'd like to talk to you a little bit about some work we're currently doing to try to tease apart some of the neuronal mechanism. So this is a brain, not necessarily on drugs, but this is the mesolimbic dopaminergic system, and this is a primary pathway for drug abuse, dopamine. And dopamine is produced in the ventral tegmental area. It goes to the nucleus accumbens, the hub of the reward pathway, and then it projects all over the brain, including the prefrontal cortex. So this is a primary pathway for reinforcing effects of drugs. So not surprisingly, a lot of the work trying to figure out how come enrichment is protective has focused here on the dopaminergic pathway. And it's really interesting. There's been some really good work done. And mostly what it's shown is there's not baseline differences in dopamine between enriched and isolated rats. However, if there's a challenge, like a cocaine injection given or something like that, there's differences in dopamine turnover or dopamine function, but baseline, there really aren't consistent differences across studies. So several years ago, we started wondering, well, if there's not baseline dopamine differences, there's only differences in response to a challenge, who's controlling the dopamine? Who is the overlord of the dopamine, therefore? So we started looking up the stream and started looking at glutamate. And glutamate is a primary excitatory neurotransmitter. And there's a long history of research with differential rearing and glutamate because it's important also for learning and memory. So all that work in the 60s, 70s, 80s, 90s, still ongoing with differential rearing and learning and memory. There's a ton of research out there that differential rearing alters the glutamatergic system to support our hypothesis. There's also work by the Collidus group and several other groups that glutamate contributes to the response to psychostimulants and the maintenance of glutamate homeostasis. So keeping glutamate levels steady within the synapse and around the synapse helps your response to drugs of abuse. And if that glutamate homeostasis gets out of whack, you're going to be more vulnerable to using drugs. You're going to seek drugs more. So our overarching hypothesis is that differential rearing is altering glutamate homeostasis really broadly. Isolated rats probably have a deficiency, making them more vulnerable to drug use, and enriched rats probably have enhanced glutamate homeostasis, protecting them from drug use. So this seemed like a good hypothesis at the time, but there are a lot of components that maintain glutamate homeostasis. There are many steps within and outside of the synapse that help maintain glutamate homeostasis. So we started looking at a postsynaptic receptor. This is a homemade glutamate receptor, if you can't tell. Okay, this is a presynaptic. We're going to release a little glutamate. It's going to bind postsynaptically. And then located postsynaptically is this metabotropic mGluR5 receptor that has a long history in learning and memory, is important for excitation and plasticity. So we started looking there, and my former grad student, David Arndt, observed that yes, differential rearing does alter mGluR5 function in support of our hypothesis, and life was good. And then we looked presynaptically at another metabotropic receptor that contributes to glutamate homeostasis, mGluR23. And this is a cool, if you're into receptors, this is a cool autoreceptor. It helps regulate glutamate release, so it can actually decrease glutamate release. It contributes to what we call glutamatergic tone. It helps maintain the amount of glutamate in the synapse. And it made a lot of sense that differential rearing would alter mGluR23 function. And Dave had an earlier paper that suggested it did, but we've run since then a series of studies with self-administration, and it looks like not so much with self-administration. So we're preparing those results now for publication, but it looks like our effects in differential rearing are much more robust here than presynaptically. So we're working on getting those results prepared. So then what we did, those are within the synapse itself, 2 key contributors to glutamate homeostasis. Then what we did was we journeyed a little bit outside the synapse to the yellow blob here. This is a glial cell, okay? And glial cells provide support for the neurons. And this is actually an astrocyte, believe it or not. And astrocytes are really cool. They provide support, both functional support and metabolic support for neurons. They also contribute to glutamate homeostasis. They've got several different receptors and transporters on them. And this guy is called GLT1, and it helps keep glutamate in the synapse. So it helps contribute to glutamate homeostasis. Okay, so there's an antibiotic you can give to alter the function of GLT1. So we were like, great, we'll work on this. And my former grad student, Eric Garcia, was, he was a graduate research assistant on this project. It wasn't his dissertation. He's gonna give the antibiotic, look at GLT1 function, and then he did some Western blots and looked at GLT1 and protein expression, and all is well in the world. Well, it turned into a 6th study, behemoth for poor Eric, because it turns out, as he was working on this paper, we learned that some other papers came out. Most of the work that had been done on GLT1 and glutamate homeostasis was done with cocaine. And as we were working on this, a paper came out with methamphetamine, suggesting amphetamines may interact differently with GLT1. And it turns out our results really replicate that with amphetamine. Amphetamine interacts differently with this transporter, and we had to take a lot of different twists and turns. So we did not find a robust effect of differential rearing on GLT1, but our results are a little inconclusive because we were using amphetamine. I'll leave it there. So, but we were, Eric got published, a great job. It's a lot more than poor Eric expected. He had some kind words for the project. Okay, so what we're doing now, or give us all hope, is we've moved also on, this is also an astrocyte. There's another transporter on the astrocyte called XCT. And XCT is pretty cool in that it exchanges an amino acid for glutamate. It actually produces about 60% of the glutamate in the synapse, which is, I was really surprised when I learned that because I thought all glutamate came from neurons, but it's actually being made also in glial cells, which is, I think, pretty exciting. And so right now, what we're doing in the lab, we're focused in here on XCT, also on the astrocytes. Why are we focused on this? There's another drug called N-acetylcysteine, which you can buy in a health food store. If you have any friends who are like into bodybuilding, they might be adding it to like their protein shakes. It decreases free radicals and helps muscle development. So you may have seen it like JNC and stuff. What they don't know, you can now tell them this. And also, it stimulates GLT1 expression, the transporter we were looking at. And it also interacts extensively with that XCT and increases that glutamatergic tone. It improves the glutamatergic tone and improves that glutamate homeostasis. So we're zooming in here on XCT and using N-acetylcysteine to alter XCT function. And I'll show you a little bit what we're working on right now to test some of our hypotheses. Due to the way amphetamines interacting with glutamate homeostasis, we have to give the amphetamine using self-administration, like I talked about. We also have to give the amphetamine via injection. So we have a couple of studies going on. In this one, we're going to inject the amphetamine. The rats don't get a choice. We just give it to them. And then we're going to administer the NAC. And I have 2 undergraduates leading this project. Jared Rack is a senior applying to medical school, and Joanne Gomendoza is a junior, psychology and pre-med. So what they're doing, they are doing 8 sessions where the animal gets an injection of amphetamine, and then they get placed in a locomotor chamber, and we just see how much they move. And again, the animal doesn't get a choice. They get amphetamine or saline, get put in the chamber, they get the same injection every day. They then get 14 days where they go back to their home cages, they stay in their home cages, and they get NAC or saline injections every day. Okay, so 2 weeks of NAC treatment. We then bring them back to the locomotor session, and they get a final drug-free test session. We give them a saline injection, put them in the locomotor chamber. We then, immediately after the session, remove their brains. It's a normal day in the lab. Then we quantify protein expression for GLT1 XCT, and then something I haven't mentioned before, GFAP. And that's a marker for those astrocytes. That will let us know how many astrocytes there are present that those glial cells are interested in. And we're looking in the nucleus accumbens, the center of the reward pathway, and the medial prefrontal cortex. So this study is ongoing. Jared and Joanne are almost on the behavioral part, and they have about half of their Western blots done. So I'll just show you a little bit of their data so far, what we're seeing. It's much more encouraging than the previous project. So right now we're in the medial prefrontal cortex. So medial prefrontal cortex in our average rats, our standard house rats. And these are the rats, these rats were given amphetamine during training. So they got amphetamine every day during training, and then they got NAC or just control during the 2-week abstinence period. So these are the control rats. This is expression of XCT, how much protein expression there was. And this is how much they traveled when we put them in the locomotor chamber. And there's no relationship. It's not significant. So no relationship between XCT expression and movement. And you would expect that, what we're seeing there in the medial prefrontal cortex. Life is good. What's interesting is after we gave NAC treatment in our standard house rats that had received amphetamine, we actually see a negative correlation with more movement, less XCT expression. We're surprised by this finding. It's not fully powered yet. They only have 5 of the Western blots done. We have 8 in the condition, so we'll see. Oh no, 6 Western blots done. We'll see how it holds up. We have all our animals in there, but that's an interesting finding to date. We did not expect that. We would expect actually quite the opposite. So we'll see how to interpret this as it rolls out. So that's in our standard house rats. What's really exciting in our isolated rats, now we're in the nucleus accumbens, the center of that reward pathway. We see a negative correlation, a really nice, strong negative correlation between XCT and distance traveled, suggesting in our isolated rats after amphetamine exposure, there might be a change in XCT functioning in support of our hypothesis. And then following NAC expression, there's still a negative correlation, but it's not significant. So again, this is not a completed study. We're still running the Western blots. They still have to finish things up, but we're excited by our preliminary results to date, suggesting at least we're tracking now closer to the right transporters and we're getting closer to where we should be going. Okay, so this is non-contingent injections. The other project that we're running simultaneously in the lab is self-administration, so contingent amphetamine with NAC. With me? It's a lot, okay. So this project's being led by my lab manager, Ted Mosier, and my new grad student, Troy Fort. He just is halfway through his 2nd year. So they did 12 self-administration sessions, and these are the long 6-hour sessions. This is actually the data I showed you. Okay, so the 12 extended access sessions. They then had 2 weeks, same model, 2 weeks in their home cages with NAC or saline injections. Okay, and then return to the operant chamber for that relapse test I showed you, and then again we took their brains. Okay, so same model, but now it's self-administration. So here they're choosing to take the drug, and then we're administering the NAC, and then quantifying the same targets after. the test session. So I showed you the acquisition, the extended access data already, but I'll show you what we have to date for our relapse test. Again, this is not fully powered yet. As I mentioned, we're running males and females. We're new to running males and females. We previously, like a lot of other labs, only used to run males. And we saw no differences between males and females during training. But we are seeing robust differences between our males and females during the relapse test, as Rick is vigorously shaking his head. So these are our males. So this is the rats who got the vehicle during the 2-week abstinence period. Our isolated rats appear to be having more relapse than our enriched rats, consistent with our hypothesis. And then following the NAC injection, it's decreasing it in our isolated rats, which is exciting. Not in our standard housed or our enriched rats. But we definitely, in our female rats, we're getting much higher rates of relapse in our control condition. And then in our standard housed rats with the NAC, we're actually seeing something quite odd where we may be seeing, it's nothing is significant, but we may be seeing almost an opposite trend where the NAC is increasing relapse. But this is not fully powered. Right now there's 6 animals in that. So it'll be interesting to see. There's very little in the literature with NAC and female rats, so it'll be interesting to see as we finish up the study what direction where this goes. But encouraging data on our isolated rats. It'll be interesting to see in our female rats. And we, not to get too into the weeds here, but we were not monitoring their estrus cycle. We did not anticipate this robust of a difference on the relapse test, but I think we will have to move to starting to at least monitor on the relapse test so we can get a better handle on some of the variability we're seeing. So anyway, so that's where we are with that. And then on our outcomes, looking at neuronal outcomes, we're quantifying the GLT1 and XCT protein expression, as I mentioned, as I showed you with the locomotor study, but we're also looking at the GFAP, the marker for the astrocytes. And to do that, we use a different technique called immunocytochemistry. And again, my grad student Troy is doing this work. And to do this, you slice the brains and then you look at them under the microscope to get a better measure of what the astrocytes are looking like. And we can count the astrocytes. We can also measure the complexity of the astrocytes. So Troy is working on this project and it's ongoing, but just to show you what we're seeing so far, he's in the nucleus accumbens, and in the nucleus accumbens, there's 2 components. There's a core and a shell. In the core, very interestingly, in the rats that have received amphetamine during training, but got the vehicle during the abstinence period, our isolated rats have a lot of GFAP expression compared to our enriched rats, and the NAC is increasing the GFAP expression in our enriched rats. Not in the isolated rats, but they're so high, could it increase it anymore? And in the shell, we see a very similar pattern in our vehicle group, which is good. It will help me sleep at night, but a different pattern for the NAC. So again, it'll be interesting to see how this pairs out. And again, we may have to also here account for the sex differences because he's taking these after the relapse test, unfortunately for poor Troy, because he was hoping not to have to process all the brains, but we're going to have to process them all. So it's good though. So that's where we are with this project. So exciting project, ongoing, helping us understand what's happening with glutamate homeostasis in our differential rearing. So to summarize, I know it's a lot of stuff. Broad picture, there's a relationship between NAC treatment and XCT expression in the medial prefrontal cortex and nucleus accumbens in our IC and ST rats with non-contingent amphetamine, more to come. And then NAC treatment during the abstinence period in our self-administration model is reducing relapse in our IC rats. We feel pretty confident to say that. And it suggests together, we feel pretty confident with these 2 experiments that XCT function may be altered by differential rearing. So we're going to continue to pursue this, and we'll be interested to see where this goes as we finish up these projects over the next year. So if you'll hang out with me for a little bit longer, you get to hang out a little longer, we good time-wise? Okay. I focus really on the wanting component, but I do want to take a few minutes to talk about another component of incentive motivation. This was a lot of time on wanting, but for an individual who takes a drug, you have to want the drug, right? You have to lever press, although we don't lever press in real life. But then when you lever press and you consume it, you have a choice to like it or dislike it. And one of the issues for individuals once they become addicted to a drug is they often want it, but they no longer like it. When you talk to them in interviews, when they're far into their addiction, they often self-report high levels of wanting, but not as high levels of liking. So we know enrichment decreases the motivation for a variety of drugs of abuse. This is really well established. I hope I've shown you some data to support that. But there's very little out there in the literature than we could find examining if enrichment alters liking. And something that's troubled me for many years is with the operant chamber, I can't tell, are my enriched rats lever pressing less because they don't like it as much, which is what I hope. I really hope it's a protective model. Or because they have differences in dopamine function, are they maybe getting high faster and they don't need as much of the drug? So I really wanted to examine this liking question, and this is what I'm doing with my Cobra project. To be a project lead, I had to learn a new area, so this is my new area. And to examine liking, I'm using taste reactivity, which is an older model established in the late 1970s, where you measure oral facial responses to a range of substances. And it's a well-established model. I'll explain that in a minute. I'm working with Stephen Kiefer, emeritus faculty member. This is what he did for 30 years. He's been a great mentor. And this is also work done by Berridge. If you're familiar with Berridge's work at all, you're familiar with this. So if you're wondering, can I tell if someone likes something from the oral facial responses? This is my son, Liam. And he's pretty horrified I'm showing this because he's now 8 and he feels strongly about this. But anyway, you may remember many years ago, the lemon challenge on YouTube. Okay, so we did being good parents. We did the lime challenge with Liam. And I think the sound's on. This is really loud because we're in a Mexican restaurant and my daughter, who's a little older than Liam, is being really loud in the background. She's not getting attention, but watch Liam's face when we give him the lime and decide if he likes it. Okay. So does Liam like the lime? No, right? It's really easy, right? You don't need any training. He hates the lime. You can tell that. And so, much to Liam's chagrin, you can also tell this. Oh, I didn't mean to restart the whole chaos in the Mexican restaurant. You can also tell this in a rat, okay? But we all think Liam's cuter than a rat. We assure him of this at home. So, in a rat, we implant an indwelling fistula. And we can infuse a small amount of liquid onto the tongue of the rat and then measure the oral facial responses of the rat. And these have been classified by Grillen Morgan, Berridge, and others as liking reactions or disliking aversion reactions. And this is actually a dislike. This is a gape. And he's in this chamber with all these mirrors because if they dislike something especially, they tend to turn away and move around a lot. So the mirrors, we video all the sessions and we have to score all the oral facial responses. So the more mirrors we have in the chamber, the more we can score. These are 1-minute trials. We score frame by frame, and each trial has 3,600 frames. So if you're a graduate student and you see, oh, this seems really cool, don't. Back away. Back away really slowly. No. So there's a lot to like, but the scoring. Okay, so again, these have been classified by other researchers as aversive. I'm not showing you all, and there's a bunch of responses, 14 total, but gapes and forelimb flails, they want to get it out of their mouth. These are aversive responses versus hedonic responses. This is a tongue protrusion. He's trying to lick it into his mouth. He likes the substance. And there's a whole bunch of ingestive or liking responses, mouth movements, and tongue protrusions, okay? So really simple experimental design. This hasn't been done in differentially reared rats. So we reared our rats in different environments. We implanted the indwelling fistula. Following recovery from surgery, we did taste reactivity trials. This, by the way, is not a real rat. This is a sock made out of a sock. We used them to set up our camera, so we had all our angles right and everything. So just to be clear. And then we infused a variety of substances because we wanted to do a dose range to ensure we caught since they're dose-dependent effects. So we did low to high doses of sucrose, low to high concentrations of sucrose, 2 concentrations of quinine. Quinine is a bitter substance found in your tonic water. They should really dislike that. That's sort of a controlled substance for us. And then 5 concentrations of alcohol. So, and going on the concentrations here, really from a Smirnoff Ice up to bourbon. And all these animals are drug alcohol naive, and they're 1-minute trials, 1 trial a day, and it's in a counterbalanced order. So the rats get them all in different orders. So lots of Latin squares designs going on in this project.
The ethanol is just diluted in distilled water, though, right?
Yes, yeah, not, when I say Smirnoff Ice, not diluted in sucrose. Yeah, no. And there has been work done as you introduce the sucrose, looking at that, but we did not get into that yet. And students in charge, this actually just got accepted for publication in Psychopharmacology, which we're 3,600 frames per trial. We're pretty excited about. And students on this project are my undergraduate Emma Brace, who's applying to grad school in neuroscience, and my grad student TJ Wookich. And we had male and female rats in this study because that hasn't been looked at much either in this paradigm, and there were no sex differences, which we are grateful for with the 3,600 frames per second. Okay, so what we did, we scored all the oral facial responses, classified them as aversive or hedonic or liking responses, and then we just looked at them at each substance to see what we saw. So looking, just to show you a few, for alcohol, aversive responses. Enriched rats lever press less in operant chamber for alcohol. They want it less. We assumed they would show more aversive responses. That makes logical sense. In the universe, these are the 5 concentrations of ethanol, aversive responses, and thankfully the enriched rats did show more aversive responses than our isolated rats, consistent with our hypotheses. And this down here just means as the ethanol concentration increased, they showed more aversion, and that makes sense. If you never had alcohol before, 40% is pretty substantial. You would also show some aversion. So we would expect that to happen. This is good. We saw a similar pattern for quinine. Where our results were very surprising were ingestive or liking responses. Ingestive responses for alcohol, enriched rats lever press less for alcohol. We would expect them to like it less. They just showed more aversive responding, right? No, of course not. So this is total liking responses are increasing concentration of alcohol. At each concentration, enriched rats showed more liking responses than our isolated rats. This was really surprising to us. And again, adequately powered, very well, all the things here. So, okay, well, it's alcohol. Alcohol is a weird substance. Let's look at sucrose. Tons of literature. Enriched rats lever press less for sucrose across so many labs. Of course, they're gonna like sucrose less, right? Why would they? So these are liking responses for sucrose. We tested 3 concentrations of sucrose. Enriched rats liked the sucrose more than the isolated rats. Liking increased as we increased our sucrose concentration. We would expect that to happen. This is good. Sucrose is good. Rats like it. We like it. But enriched rats like the sucrose more than the isolated rats. All right. So I'll explain what we think is happening in a minute, though. All hope is not lost. It's actually pretty exciting. So to summarize, when compared to isolated rearing, enrichment increases ingestive responding, which we did not expect, but we did hypothesize would increase aversive responding for alcohol and quinine. But when we looked at the data as a whole, this is the quinine. I didn't show you that, but it replicates the ethanol. We were pretty overwhelmed by the size of this project with the scoring and 3,600 frames per second. There was a lot. It was a lot more comprehensive than we thought. But as we looked at the data as a whole, we noticed that on the liking responses in particular, our isolated rats, they were just flat on the ethanol and almost flat on the sucrose. And we analyze these using mixed effect models. We have regression on them, and very, overall, very flat. So starting to think about it, they seem really insensitive to the changes in concentration. They're unable to detect the liking, which then is actually quite an exciting lack of support for our hypothesis. So now we're hypothesizing that maybe isolated rats have a shift in their hedonic set point, and that may explain their increased wanting behavior. they're unable to detect the liking. That's why they keep lever pressing. That's our current hypothesis. So we're actually really excited now. So isolation rearing appears to shift the hedonic set point. They have a overall reduced responsiveness to rewarding stimuli, sort of they shifting the allostatic set point. They look like a rat exposed to extended access to a psychostimulant, but they're drug naive. They look like that drug user who shows high levels of wanting, not liking, but they're drug naive. So we may have a unique phenotype here that we're pretty excited about. And it may explain the increased wanting behavior. And we just submitted a small grant, R21, to follow up on some hypotheses about this that I'm happy to tell you about later if you're interested. So that's where we're at with this. So what we're doing now with the COBRE project, though, these were planned before we had our results, and this is still exciting too. We're administering alcohol to the enriched, isolated, and standard house rats during adolescence, and we're seeing if we can shift liking and wanting. So we are looking at taste reactivity after alcohol exposure during adolescence and operant responding after alcohol exposure during adolescence. And then with my colleague, Stefan Bosman in chemistry, we're actually doing diffusion tensor imaging, and we're looking at the neuronal microstructure integrity, because alcohol alters the integrity of the white matter, and we're interested if enrichment is going to protect against that damage to the white matter. So we're in the middle of this project right now. Happy to talk a little more about that. And then also with our group over in chemistry, we're also doing nuclear magnetic resonance imaging to quantify glutamate and several of its metabolites to see if there are changes in glutamate function as well with this project. So that's where this project is going with the COBRE project. So summarize, there's a lot, take two bodies of literature, but hopefully coming together. Take-home points, differential rearing results in changes in drug taking and glutamate homeostasis. And this change overall is resulting in protection for enriched rats. Isolation rearing results in an insensitivity to reward magnitude based on our taste reactivity studies. And we're working now with our colleagues in chemistry to determine if this is related to changes in glutamate. We'll see what happens. And then take home, definitely differential rearing is altering both the wanting and liking components of incentive motivation, but perhaps not in the directions we first anticipated. So thank you for your patience. I know that was a lot. Thanks for hanging in there on Thursday afternoon.
Question?
Yeah.
So the first bullet, differential rearing, I assume that it's known that doing that late post the initial however many weeks doesn't have any protective benefits. Like return, and if you were to do the relapse test and take the isolated rats and put them in the enriched state in between the priming and the relapse test, there wouldn't be any benefit to doing it late.
Actually, that's a great question. Is there a benefit of enrichment later? And there still is. Actually, Beline and Salinas and some other groups have asked that exact question and placed them in enrichment during adulthood, during the abstinence period, and it does decrease self-administration. No one has yet compared directly within a study, does it increase as much as placing them in during childhood, but it still offers protection, which is exciting. Is it as good? We don't know, but it does offer some protection, which is hopeful, definitely hopeful. Yeah, Ken.
So your last data was actually quite fascinating. Great work. I'm looking forward to the publication.
Thank you. Don't 3,600 frames a second.
No.
Remember.
But what really interested me is its original conception of hypothesis. I was wondering if we could discuss this a little bit. But while it's true that even Parrish and Robinson say this, that normally we consider liking and wanting to running in parallel in the same direction, but in some, and that's what we used to think, that it was one system. But I think perhaps it's better to conceptualize it as two parallel systems. And so it could be true that the social animals are more reactive to the hedonic reward, but that they like more. But if that system is parallel but not the same as the wanting component, maybe that's why you're seeing less wanting. Yeah, it's sort of like the inverse of your hypothesis. It should also be true, instead of changing a hedonic set point, these systems could be completely, actually, in this situation, separate.
Yep.
And so I guess how to design experiments to tease apart that question is going to be interesting.
Yeah, that's a great question and comment is, are they separate systems? And that's actually when we wrote the original proposal, that's how we wrote our alternative hypothesis, was that they were separate systems. And that may still indeed be the case. And that, in terms of testing it, it's going to be, if that is the case, it'll be a long road, which is good in terms of funding. If we're on the right track, it could be a long, good road, but it's also going to be hard. Do we want to be scoring all this? So my students are like happy and agitated at the same time. But anyway, but no, you ask an excellent question: is how are we going to have the right?
Yeah, that's a sort of data, right?
Yeah.
Want but not like this.
Yeah, I will say that too. Then, how do we design the appropriate behavioral controls to answer your question? That's the $1 million. Yeah, that's going to be the hard part for us.
Doesn't Aaron Edberg have some data to that effect? Because he uses that runway model, right? Or what happens is a rat that repeatedly self-administers a lot, they'll run like hell down that runway, like, I'm going to get my drug. And then when they get to the goal box where they're going to get the infusion, they stop. And they're like, they don't enter as fast. And so there's kind of a dissociation between how fast they'll run, presumably the wanting, and then the entering, where you can actually get it. Yeah. So I was wondering, have you thought about that?
In this model, we talked about it and shied away from it because enriched and isolated rats differ in their locomotor behavior. But
That's fair.
in a standard housed rat, yes, that could be if our hypotheses are supported as we get further down the line, yes, we could use that as one of our tests. But in this model, we have an unfortunate confound. No, but that's a great model. We got all excited initially.
You typically work with long Evans rats or Sprague dollies?
Yeah.
I saw one of them, there was like pictures of Sprague dollies, and in the second piece is all long Evans.
Yeah, great catch. With our psychostimulant work, we work with Sprague dollies. With the taste reactivity work, we're working with Long Evans because the second part of the project, we're giving them alcohol, and they respond more for alcohol and everything. So we do both now. So our alcohol work is all Long Evans, our psychostimulant work is all Sprague dollies. And my students all like the Sprague dollies better.
I think you can get agreement from my lab too.
Yeah, they're getting crankier and crankier. Yep.
Can you talk a little bit about what you think some of your results imply for therapy treatment amongst humans? Do you think about that and how does it connect?
Yeah, it's a great question. What do the results imply for therapy treatment for humans? And, and overall, it's apparent too. Enrichment definitely is good. And I think coming to your question, does enrichment in adulthood do anything? I think the important thing is, I think if we can enrich, offer those alternative reinforcers, offer forms of exercise, anything we can do along those lines early, I think will be good to offer protection later. But I think too, it's important to step back as a preclinical researcher and say it's so much more complicated in the human world. I mean, that isolated condition, thankfully it's a rare childhood environment that's like that, right? The extreme rare case you read in the news. So what is the moderately deprived childhood environment? Where does that fall in my model? And that's, I think, a much harder question to ask, to answer. Yeah, great question.
The choice to do it with alcohol instead of following on with some of the other psychostimulant work that you had done was?
Honestly, for the COBRA, branching out into a new area. Yeah. So, yeah, and taste activity historically has been done with alcohol, but with our results, we can nicely apply it back to our psychostimulant work. So we're excited about that with the taste for activity, that it's a shift in reward magnitude. It fits nicely with our other work, so.
Yeah, it's just thinking, I mean, there's some, there's some question about whether or not there's similar reward pathways for psychostimulants, and that's sort of a question. Yeah, so it'll be interesting to see where the alcohol work goes. A big issue we've had with the alcohol work is administering the alcohol during adolescence. Because our rats are group housed, we can't allow them to drink it voluntarily because we have no idea how much they're drinking. So I'm working with a group of students in engineering, and they're working to develop a bottle we can use in our group house cage, or we can track each rat, but if they can develop it. But until that time, we're using injections, which is not ideal at all. There's a lot of problems with that. So that's something with the alcohol stuff. If we can't resolve that problem, I don't know if we'll continue or not, because we could also intubate them. We could put a tube down their throat and give them the alcohol that way. It's a little bit better than injections. But with taste reactivity, when you pull the tube out, they can get alcohol in their tongue, and that could have mucked up that part of the study for us. So. Yeah, but if those students can develop the models for us, that's going to be great. They're really optimistic they can do it. We'll see. It's like their group project for their engineering class. So awesome. Yeah, this is awesome. Yep.
I guess I'm wondering if there's a difference between the effects you see in the stimulation condition with the novel objects versus just the presence of other rats, multiple other rats. Because for the standard condition, it looks like you had two together. I'm wondering, lots of other rats, like a community of rats, would that have any effect as opposed to, a different effect as opposed to like just the novel objects presence?
Yeah, some groups have tried to tease that apart, the question being, so what if you had a group cage with just a group of rats with no novel objects? Is that as effective? And in general, you want all the components. You want the handling, the objects, and then the cohort all together. So the group is better than being alone. The group is better than two, but you want the group handling and objects for the most robust enrichment. But yeah, some groups have done really nice systematic studies where they put 2 rats together, 4 rats together, 8 rats together, and then they add objects and see what's the best outcome. Yeah, good question.
So the first piece, you saw that during the relapse, the reinstatement, the increased reinstatement, you saw that enriched rats in the extended access had this protective factor, right? And then also the short term as well. But is the relapse mediated by the lack of sensory deprivation for those, isolated rats, and not by the motivation to seek drug, it's the motivation to seek the chelate?
Yeah, that's an excellent question. Is the increased relapse in the isolated rats due to the wanting of the sensory of the light? Is the light itself reinforcing? I would argue that's definitely a component. We haven't teased that apart. That's a great question. That'd be a great project for a student. Yeah, I think we've done some work based on Rick's work where we've just had isolated rats pressed just for a light continuously, and then we tried to extinguish that light for 25 sessions, and they still would lever pressed even in the absence of the light. But I think we could do something maybe where they just lever press for the light, then we do an abstinence period and see if they just lever press for the light during a relapse test. Yeah, that would be a fun
I wonder, like the
side
first
project.
abstinence period, because there's no light, there's no opportunity for that kind of enhances that maybe visual stimulus reinforcer when that time comes for the relapse.
No, that's a great question. Yeah.
So I have a question that might not make sense if the first question that I asked isn't true. So when you have the rat that takes in whatever substance and they get put back with their cohort, is the rest of their cohort, have they also had a substance, or are they essentially sober?
Most of the time, we try really hard that everyone in the cage has the same substance. So a lot of our rats are also getting saline, but we normally have 2 cages, a saline cage and an amphetamine cage, or a saline cage and an alcohol cage. So most of the time, everyone's drunk and everyone's high in the same accordingly. 90% of the time, we have that world.
Okay. Well, the reason I asked is my second question is, if there's any way to measure how the other rats respond to the rat that gets put back in if they are not sober and the other rats are sober, in terms of like discrimination or how they face, you know, what that interaction looks like and if it changes and if that somehow makes a difference in wanting and liking.
Yeah, it's a great
Has
question.
anyone looked at that?
Just a little bit of data with opioids actually recently
Okay.
around social media, and it does matter.
Does it?
Yeah, so actually it does turn out like if you're roommates, they've not done it here, they haven't done it here
So
in
if you're
complex
in work
systems.
setting.
But
you have
if
a
your roommate is stoned, comes home stoned quite a bit, and then you have finally an opportunity to get stoned, you're more likely to get stoned than if your roommate doesn't come home stoned all the time.
Fascinating.
Yeah, they don't know the controlling variables yet, but again, it's not
Cool.
the complexity.
No, but that's
You know, 10
interesting.
drunk rats all flopping around because they're hilarious.
Yeah, because I know they've done some of that work with fear conditioning, where
Right.
the fear from the one rat will
Right.
kind of transfer people to the other rats, but interesting.
I love the direction you took with the alcohol. It was not what I was expecting. What I was expecting you to do was to bit parallel more like what Linda Parker did, right? Because she does like taste aversion with the amphetamine, so she'll take, or with another drug. and-
3,600 frames.
No, I know that, but I was like applying it over to the amphetamine, right? Does it parallel?
Yeah,
Right,
no,
you could do this, pair the saccharin or sucrose with a high dose amphetamine because, as you know, the taste aversion looks different.
mhm. Yeah, it takes us about, yes, where we're really stuck right now in the lab, it takes about 45 minutes for us to score each trial. So we're really, there's a lot of projects we are designing that we have ideas for. And we're working with a group in Brown to use a machine learning algorithm. And they were super, they're great. They've developed machine learning algorithms for other groups, but the mirrors are really causing problems for them because they can't train it to know our rat smell from the images in the mirrors, and we can't take away our mirrors. So we have to figure out, yeah, there's some, we also want to ask some other really interesting questions we thought we could ask, like with regarding caffeine and alcohol, some really basic, like that my undergrads are really interested in too. And but the scoring really actually slowed our whole lab down with the 45 minutes per trial. But yes, we could ask some of those great questions like she did. Yeah.
This is an awful long question.
Yeah, that's what we're trying to do with our group, with the group at Brown. They've now had over 100 trials for 9 months, and they still can't develop an algorithm because of the mirrors. I hope so, because we're really excited if they can. We would love that. Yeah, we worked with a group at K-State, and they couldn't crack the nut, and we sent it out to the group at Brown. So we'll see what happens. I don't know if you know any good machine learning people who want a behavioral project. Yeah. Everyone gets really excited about it, and then they see the mirrors. And
Thank you very much.
Yeah.
thank you.